Today I'm going to talk about iCn3D visualization, analysis, sharing of protein structures and interactions. Oops, sorry. Okay, so first I like to compare different 3D viewers. There are web-based like iCn3D, Monster, NGO, and Aquario, and also standalone applications like Camera, Pymo, and iCn3D. So most of the 3D viewers show the 1D information, which is a sequence. And some of them also show the 2D diagram or kind of 2D display. And, okay.
I'm sorry.
So one unique thing about iCn3D is you can share your results. I think I go back. How to go back? How to go back to the previous one? Okay. So I highlighted here. So this is unique about iCn3D. Once you generate your custom view, you can output as URL, which typically is a short URL, and you can share with your colleague or publish in your publication. And since annotation is important, so most of the viewers have annotations either in the software or through integrated website, and like alignment feature are available in iCn3D, Monster, Aquario, or iCn3D. And iCn3D, you can process many structures in batch mode using Node.js script or Python script. This is available in Pymo as well using Python script. So NGO and iCn3D can be used in Jupyter Notebook, which is kind of, very handy sometimes. And iCn3D can provide 3D printing and virtual reality. Some like Camera I think can do both. Aquario has some virtual reality feature. Okay. How to go to next one? This one. Cursor. Oh. Okay. So here I shows one example of iCn3D showing the integrated 3D on the left, 2D in the middle, and 1D sequence on the right. Right now it's showing the summary of the sequence. So in the 3D, it detects IG domains in this structure. There are 3 IG domains, and it's colored by IG domain with different color for different strands. And 2D diagram shows that, like, square shows the nucleotide, circle shows the protein, and diamond shows the chemicals. And the lines between the nodes shows the interactions. So you can click on that to show the synchronized views in 3D, 2D, and 1D. And I'd like to focus on the 1D. Here shows different kinds of annotations, like conserved domain, CleanVas, SNPs, functional site, post-transcription modification from UniProt, and transmembrane and IG domain information. So if the annotation is available in your protein, if you click all, it will show up automatically for your protein. So that's a way to cross-compare for each residue. It's involved like binding site or domains or interactions. So just one click, you can get all the information, annotations. Here shows a 2D cartoon of PLC beta 2. So this is different from the 2D diagram I just showed you. Basically, here it's a 3D projection, actually 2D projection of a 3D structure. So if you change its orientation, and the 2D projection will be different. And you can also move around the nodes. So this might be handy to generate kind of 2D cartoon for your structure. And the line is basically it's a connection between the different domains. So this one has the pH domain, EF hand, catalytic domain, and C2 domains. And these are shown as part of the conserved domain in the 1D tracks. Now I'd like to show some interaction between the spike protein and ACE2 in two ways. The left one shows a 2D interaction network where the two structures are shown in two lines. Basically, I mean, two sets of the interaction partners are shown in two lines. The pink one is actually ACE2, I think. The blue one is spike protein. So the line between the residues are the interactions. They are colored based on like different colors for different interactions. The green is for hydrogen bonds, and gray is contact. And on the right shows the 2D interaction map. And some people might prefer the map because it's not so messy. So the two sets of residues are on X and Y axis, and the interactions are the points in the graph. You can also do mutation analysis in iCn3D. For example, the N501Y mutation in spike protein. You can show the, here shows a mutant structure. So the asparagine was mutated to tyrosine. And then here shows the different interactions. Basically, you introduced one pi cation interaction and one pi stack interaction. And in the sequence and annotation windows where all the annotations are showing, you can mouse over to the residue, show the SNPs, different SNPs. In iCn3D, you can, with one click, you can show the delta electrostatic potential. It's done by the delta program licensed from Barry Honig's group. So the left part shows the ribbon diagram with the red indicates negative charge residues, blue indicates positive charge residues. And the middle part shows the potential on the molecule surface. So if it's less than minus 50 millivolt, then will be red. If greater than 50 millivolt, will be blue. But on the right, it shows the electric potential profile. Basically, on the profile, red will be minus 50 millivolt potential profile. Blue will be positive 50 millivolt. So this is accurate potential profile, but on the surface, it's a range. If it's above 50, it shows blue. There are many alignment features in iCn3D. It can be used for both alpha-4 structure and PDB structures. So you can start with alignment, or you can load your structure and then do realignment. And alignment can be between protein complexes using VAS+, or you can align the chains in the structure using VAS+, using VAS or TM-align. And alignment can be based on structure or on sequence or residual mapping. Basically, you define the alignment. So here shows one example of the, I think Aaron showed this, show the VAS alignment of one PDB structure with two alpha-4 structures. So the left part is done by VAS alignment, and the right part shows the sequence mapping based on the structure alignment. So recently we developed, collaborated with Ravi and Philip, we developed an IG numbering system called IG strand reference number. Basically, it can detect IG domains in any input structure and assign reference number for each strand. So this is a CD8 IG domain. From N terminal to C terminal, it has the strands from A to G with different colors. And I like to introduce the schematic style in iCn3D. Basically, the sequence is put on the structure. So for example, the IG domain starts with QFR, which also labeled on 3D structure QFR. So it kind of combines the sequence with the structure. This works well for like simple structures. Okay, almost done. So basically, you can use iCn3D in different environments. You can use it in web browser and using a shareable URLs to communicate your results with colleagues. And you can process a large set of structures using Node.js script or Python scripts. And iCn3D can be used in, it's available in NPM. You can integrate into your projects. It can be used in virtual reality and augmented reality. It can be used for 3D printing as well. Thank you for your attention.
Thanks, Jiyao. So we have time for a question or two.
We have a workshop at 3 to 7:30. So
no, 6 to 7.
Oh, 6 to 7:30.
Yeah, so
yeah, Zhao, I have a quick question. So obviously iCn3D is a tool that you push updates to a lot. I was just wondering what is next and what are some things that you're working on that we might see soon.
Okay, so annotation is one of the major area we're working on in iCn3D. So it integrates annotations from NCBI, from EBI, and in the near future, we also try to like add more information to the interaction analysis for chemicals, so we can show the chemicals as 2D diagram and show the interactions for each atom in the atom level. I think that's actually done in the EBI, but we would like to do something similar in iCn3D as well.
All right, very cool. Thank you very much.
Thank you.
all right, yeah, thanks, Jiyao.